Okay, this is a nice paper that reports the analysis of all the data in the world on how the so-called stoichiometry or elemental ratios, or the more famously known Redfield ratio, is changing across the oceans and the coastal regions and the open ocean and near the surface and deep below the surface, etc. It says global scale shifts in marine ecological stoichiometry over the past 50 years. If you don't know what Redfield ratio and stoichiometry are, basically there are ratios between carbon, nitrogen and phosphorus. Sometimes iron is also included. And why is it important? Because it was found, I think, in the 1930s by Redfield that the ratio in the cells of the phytoplankton and in the water were very close to each other, raising questions about how this happened, whether the phytoplankton started to assimilate nutrients at a certain ratio, and over time that began to reflect in the waters, or does somehow ocean waters have ability to maintain this ratio, and so on. But they have importance because with human activity in the coastal regions, for example, we are increasing the nutrient runoffs, which can change the C to N to P ratio or N to P ratio and so on, which affects the species composition. There are large phytoplankton, small phytoplankton. You can also do classification by functional types, Prochlorococcus, Synechococcus, Diazotropes, this and that. But this study basically sees whether those ratios are still the same as was found way back, well over half a century ago. Let's read the conclusion first, because I'm going to be brief on this. Our study reveals substantial global deviations from the classical Redfield ratios, with marine C to N to P, carbon, nitrogen, phosphorus stoichiometry varying markedly across depth and time. We'll see a figure. These ratios have not been fixed across the world anyway. Within the Mediterranean, they show a large range, for example, because there are many factors that control this ratio, as we'll see in a minute. These findings underscore the dynamic, non-static nature of marine elemental ratios, emphasizing the need for next-generation biogeochemical models that incorporate spatial heterogeneity, flexible stoichiometry, and temporal feedback. So models cannot just assume that they are fixed, which we often do. Sometimes models use that as make that a prognostic variable, but that's not easy, because you are either carrying nitrogen as a currency or carbon, and then when you carry nitrogen, you are using stoichiometry to produce carbon numbers on primary production, export, and so on. Okay, understanding such variability is crucial for predicting how marine ecosystems will respond to accelerating global change. Okay. Let's read some text that is useful. I'm being very brief, so I'm just taking bits and pieces. The elemental stoichiometry of carbon, nitrogen, phosphorus regulates marine biogeochemical cycles and underpins the Redfield ratio paradigm. However, its global variability and response to environmental change remain poorly constrained. Here we compile a global data set of 56,031 plankton or particulate matter that's collected in by various ways, nets and sediment traps and so on, and 388,515 seawater for dissolved samples from 1971 to 2020, spanning surface to 1,000 meter depth, to assess spatial and temporal dynamics in marine C to N to P ratios. We show that planktonic C to P and N to P and oceanic C to N and C to P ratios consistently exceed Redfield ratios throughout the study period, indicating widespread deviations from canonical stoichiometry. Planktonic C to N and N to P ratios rose markedly in the late 20th century, followed by a decline, suggesting a progressive alleviation of phosphorus limitation, probably driven by increased anthropogenic phosphorus inputs. Probably, which means it's a hypothesis. Depth resolved patterns show decreasing oceanic C to N and C to P and increasing N to P ratios. Okay, it's a complicated story. And these are attributable to differential remineralization and microbial nutrient cycling. So phytoplankton assimilate these nutrients. They may die, aggregate, and rain down, or eaten by other heterotrophic animals in the water, and they release. So the remineralization is basically organic matter back into nutrients that are inorganic, like carbon, nitrogen, and phosphorus. Okay, our findings highlight dynamic non-static stoichiometric patterns over decadal scales, offering critical observational constraints for refining representation of elemental cycling in biogeochemical models and improving projections of marine ecosystem responses to global change. So we have a bit of a tunnel vision on carbon cycle, but these things are the fundamental components of carbon cycle, so we have to worry about their functioning. They are not saying it's related to global warming, but human activities like phosphorus, increased phosphorus supply to ocean waters, are also playing a role. Marine carbon, nitrogen, and phosphorus cycling plays a fundamental role in global ecosystem processes. The elemental ratios of C to N to P in plankton as particulate matter and seawater as dissolved substances are key regulators of material transformation and energy flow in the marine ecosystems. Redfield's seminal work revealed a remarkably consistent elemental composition of marine organic matter, proposing the C to N to P molar ratio of 105 to 15 to 1 as a product of marine biological activity. So its conclusion is that the activities, biological activity, is determining the ratio in the water. The average molar ratio of C to N to P in plankton 106 to 16 to 1 and the ocean 1017 to 15 to 1 have become foundational in oceanography and marine ecology, offering useful baselines and important insights into nutrient dynamics that sustain marine life and drive global biogeochemical cycles. Okay. Just a few more points. Ecological stoichiometry in marine ecosystems is shaped by multiple factors leading to complex temporal trends in C to N to P ratios, increased N and P inputs from the atmospheric deposition and riverine fluxes combined with the relatively constant ratio of oceanic carbon uptake may contribute to a reduction in oceanic C to N and C to P ratios. So it's given in a supplemental figure you can look up. Conversely, rising carbon dioxide concentrations could increase planktonic C to N and C to P ratios by enhancing carbon utilization in marine organisms, so kind of a ocean CO2 fertilization effect, which is much more commonly discussed for land vegetations. Furthermore, regional variations in nutrient availability, in particular increasing N and P inputs in Asia, and adds another layer of complexity to these dynamics. Climate-induced changes such as ocean warming and acidification generally favor higher planktonic C to P ratios through enhanced carbon mobilizing enzyme activity and reduced ribosomal phosphorus demand. So very detailed conjecture of what are the contributors. Despite these potential drivers, the factors influencing the marine ecological stoichiometry often act in opposing directions, leaving the overall trajectory and magnitude of changes in marine stoichiometry unresolved. So they are reporting what is seen in the data, but they are offering potential drivers and not necessarily the answers. To address these gaps, we compiled an extensive data set of what we mentioned before, and they want to quantify deviations. They wanted to, when they started, to quantify deviations from the Redfield ratio in planktonic and oceanic stoichiometry, identify potential temporal trends in the planktonic and oceanic ecological stoichiometry, and develop theoretical models to predict changes in planktonic and oceanic ecological stoichiometry across seawater depths. So it's not just a surface change. The changes penetrate to the depth, and they are different from the coasts to the open water. So the first figure is showing all the observational, ship tracks and some buoys and ships stop and put sampling instruments and so on. So they measure plankton down to about 200 meters and below 200 meters or down to 1 kilometer, and also in the ocean, similarly from 0 to 200 meters and 200,000 meters, and you can see the symbols shown here, and the counts for each are shown. So obviously you expect ocean water sampling to be higher. These are log 10 counts, so they are fairly large numbers that we mentioned before. 388,000 388,515. Having a brain fade over there, but anyway, you get the idea. Here the coverage has not been uniform over time, but nonetheless you see what is used, and then you look at the ratio 0 to 200 meter, 200 to 1000 meters, and the Redfield ratio. In the plankton, you are looking at phytoplankton in micromoles per liter, and phosphorus and carbon in micromoles per liter. So you have Redfield ratio here, so there is nitrogen there, and you have oceanic phosphorus, carbon, and nitrogen as well. Okay, these are from various parts of the global ocean: North Pacific Ocean, South Pacific Ocean, North Atlantic, South Atlantic, Mediterranean, Southern Ocean, Arctic Ocean, South China and Eastern Archipelago Seas, Indian Ocean, and so on. So there is a fair bit of global coverage, so that you get a good sense of the change over the whole world ocean. Okay. Should I say something else? Look, yeah, let's leave it here. I'm going to jump ahead to the final schematic figure that summarizes the ideas. A conceptual diagram illustrating the spatio-temporal variations. There is a table with details on how things change over depth and C to N, C to P, and N to P, and so on, that I have skipped. A conceptual diagram illustrating the spatio-temporal variations of marine ecological stoichiometries of carbon, nitrogen, and phosphorus. This figure illustrates the conceptual framework of spatio-temporal dynamics in marine ecological stoichiometry for C, N, and P, highlighting how the ratios and distributions vary across spatial scales and over time. The curves showing stoichiometric ratio changes with ocean depth are adapted from from figure 3, which I haven't shown, but it comes out of the data analysis, while those reflecting temporal variations are based on figure 4, which also I have skipped. Please follow up with the paper if you're interested in this. I'm summarizing this just for kind of non-biological people. Maybe, many times people do carbon cycle without going into such details on the ecosystem side. People who do biogeochemical modeling obviously have to worry about this, because they provide not only primary production and their trends and spatial temporal variabilities, response to El Nino, etc., but they also provide the input to global carbon budgets and climate feedbacks, etc. Right? So here we are going from over to 1970 to 2020. Oceanic C to N seemed to increase, and around 2004 to 2007 or so it peaked, and then it's been declining. Planktonic C to P, oceanic C to N, planktonic C to P peaked a little bit later, around 2007. Planktonic N to P maybe slightly later, but all are declining here in terms of the ratios. So here you have low to high stoichiometric ratios going from depths towards the surface. So C to N, you can see how the ratios remain relatively constant, but if you look in the 200 meter range, there are strong gradients. So what are the factors playing a role? Clouds, atmospheric deposition, riverine inputs, oceanic nutrient cycling itself, and you have N2, nitrogen fixation. The diazotrophs I mentioned do not need nitrate. They can directly assimilate dinitrogen in the water, and you have ammonium nitrification processes producing NO2 minus and denitrification to produce N2. So you have plankton metabolism interacting with oceanic nutrient pool in terms of uptake and mineralization. So you have carbon, nitrogen and phosphorus in the plankton metabolic pool as well, and you have energy coming from the sun, which is obviously very critical for photosynthesis, which happens typically in the so-called euphotic zone, which is the depth at which the surface radiation drops by 1 exponential and so on. So CO2 goes in, oxygen is released, but respiration uses oxygen and releases CO2. So basically I won't go into the detail, but you have sedimentation and release, so downwelling and upwelling processes which connect the biological pump, the solubility pump, the remineralization, and of course help with ecosystem functioning and the C to N to P ratio over time. So N to P, you can see again how the near surface changes are much more than the deeper changes, but here you see also C to N, C to P, N to P, and you have light nutrient ratios which are important, which is high near the surface because light is available and drops off as you go to about 1 kilometer. Is this very helpful? Well, just focus on these things and how things have changed over time. And as we said, this is just a data analysis pointing to the changes. It doesn't necessarily give you the answers as to exactly how it happened from surface to depth and from place to place. Nonetheless, they have some suggestions on what the drivers could be. Okay, the main point is that things are changing, and the global biogeochemical models which use these ratios from historical observations have to worry about how to represent their non-stationarity or their changing characteristics in the model. Okay, let's leave it here.